You will use an apparatus like this one to perform small-scale dry column vacuum chromatography. You will gently insert a pipette chromatography microcolumn into the hole of the rubber adapter at the top of the apparatus. Don't use too much pressure when inserting the column. Push it just enough so that it's secure and makes a seal. The preparation of the microcolumn is demonstrated in another video. See the experimental procedure for instructions on how to load onto the silica at the top of the column the material that's to be chromatographically separated. The apparatus is connected to a vacuum source via thick-walled rubber hose. When vacuum is applied, the liquid mobile phase that passes through the column will collect in the separatory funnel. You will then break the vacuum, allowing you to drain the liquid from the separatory funnel. This process is now demonstrated. First, close the separatory funnel's stopcock valve. Then attach the pinch clamp to the rubber hose on the T-connector to create a closed system within the apparatus. Then fully open the valve of the vacuum source to create a vacuum. You can then use a pipette to add the mobile phase, also called the eluent, to the top of the loaded column. Let's assume that this is the eluent to be used for the first column fraction. The vacuum will pull the liquid rapidly through the column, and the liquid then collects in the separatory funnel. Once the liquid passes through the column and the column is mostly dry, which should take no more than about 30 seconds, you should remove the pinch clamp from the rubber hose on the T-connector. Then close off the valve at the vacuum source to stop the suction, and then drain the column fraction into the appropriately numbered test tube, in this case number 1, since this is the first column fraction. Don't forget to close the stopcock valve after each time you drain it. Note that you do not have to clean the separatory funnel or any other part of the apparatus after each new fraction is collected. You can proceed directly to adding more eluent to the top of the column and collecting the next fraction in the same manner that you collected the others.